very intense discussions with Babak and Andrew while they were working on this. And secondly, since DeepBind, we've started a company called Deep Genomics, and DeepBind is one of the core technologies that this company is built on, and I'll comment a little bit towards the end what the relationship of DeepBind to our efforts are. So after the previous two talks, I think DeepBind doesn't need that much introduction anymore. But just to recap, it's a machine learning model, and the inputs are sequences. You train it to predict a particular protein, and the output is a binding specificity of that protein to that sequence. What is very nice about DeepBind is that it's not restricted to one particular technology. In fact, you can train DeepBind both on microarray data, which predicts binding affinities directly, as well as in vivo sequencing data, which does not give you a direct measure of the binding affinities, but gives you a pool of high affinity sequences. So this is the high-level overview of the DeepBind model. The input is a sequence, and you apply a set of convolutional motif detectors to that sequence. Then you apply a nonlinearity, you apply max and average pooling, and then you feed these filter responses through a neural net, which learns a non-linear function and makes a prediction. And then, of course, you contrast that prediction with your measured targets and train the whole system using backprop. So as Howie Yang pointed out, this sort of architecture is something that today a lot of people can whip up in an afternoon in TensorFlow. But I'd really like to point out when this work was done, this was really a Herculean effort on the behalf of Andrew DeLong, because none of the tools that we use today, like TensorFlow or the QDNN libraries for convolution, were available. So all of this is essentially built from scratch, and Andrew went even so far as to implement convolutional kernels on the GPU from scratch, which is a really hard thing to do. So Andrew and Babak trained this model on both DNA and RNA binding data from both microarray assays as well as CELEX and chip sequencing data. And the first evaluation that they did was using data from the Dream5 challenge to predict mouse transcription factors. And in the left panel, you see the scores. So they trained the model on microarray data, and this is the performance on held-out microarray data. So DeepBind set a new state-of-the-art for this task. But then what kind of made DeepBind stand out was that if you took the model that was trained on in vitro microarray data and applied it to the in vivo ChIP-seq data, DeepBind still outperformed all the other models. And in fact, if you look at the two runners-up on the microarray data, these two models do considerably worse when you apply them outside of their domain on in vivo ChIP-seq data. And on the opposite, if you look at the runner-up from the in vivo ChIP-seq data, that model does considerably worse on the microarray data. And so that just gives you an indication that what DeepBind learns really seems to be biologically meaningful and is not overfitting on any kind of technological bias in these measurements. I'm not going to comment on the RNA binding evaluation in detail, but I want to point out some interesting examples. So this was similar. The DeepBind was trained on RNA-compete microarray data and then evaluated on in vivo ClipSeq data. In black, you have the performance of the PWM model in RNA-compete. And you can see that while DeepBind outperforms the PWM model on all these proteins, the difference is not that large on the majority of them. However, for a few select proteins, you have very large differences. And to find out why that happens, I'm going to look at this particular example in TAR-DBP. And what we have here, this is the PWM that RNA-compete learned. And what we have here, these are the filters that DeepBind is using. And then uses a nonlinear function of all of these filter responses to predict the binding specificity. But we can see that DeepBind learns motifs that are more subtle and learn more long-range effects than the simple PWM, and that might explain why on proteins where that might be important, it outperforms RNA-compete by a significant margin. So a common criticism of deep learning methods in biology is that many people consider them a black box and difficult to interpret. And Andrew and Babak thought very hard about how to not only get accurate predictions from deep learning models, but also get valuable interpretations. And they came up with this concept of a mutation map. The mutation map consists of two parts. On the top row, you have the wild-type sequence, where the height of the letter indicates how correlated this position is with the predicted binding score. And in the matrix below, you can see how each possible mutation is predicted to affect the binding score. So the darker the blue shade is, the more a mutation is predicted to decrease the binding score. And so here we're looking at a particular example in the SB1 motif in the LDLR reporter. So LDLR is a protein whose job it is to remove cholesterol from the bloodstream. And for example, we can predict that at this highlighted position, a mutation from C to T would lead to a loss of this of binding of this transcription factor. And indeed, when you look at all of the known disease-causing mutations from HG and D in this promoter, you see that within this motif, DeepBind correctly predicts loss of binding for every single one of these HG and D mutations. So DeepBind is available at this address at tools.genes.ca/DeepBind. On this web page, you can browse all of the filters and motifs that DeepBind learned, as well as download an executable that allows you to make your own make predictions for your own sequences. So I'd like to comment a little bit on the state of the field and this community. So as the two previous talks have shown, DeepBind has really created a lot of interest since it was released last year, and there have indeed been numerous efforts to improve on the DeepBind model. So there have been the DeepC, the Computer, and the Bassett algorithms. What they did, so they added more features to their models, did multitask training, and also used better background models for the to learn from the sequencing data. And we've seen the previous two talks in this session that showed that if you take advantage of secondary structure, as well as explore more and deeper model architecture, that will also help with your performance. So we think it's really encouraging that that DeepBind really inspired so many people to iterate on this starting point. And on the other hand, if you look at ISMB this year, you can see that deep learning applications are now prevalent in many different domains and are very quickly becoming a central pillar of biotechnology. So it seems that this community is really moving forward very, very fast and making very quick progress. However, when you look outside of the community and the academic world, you can see that machine learning algorithms are not very widely used for phenotype prediction or variant interpretation at all. In fact, variant interpretation in the clinic remains a very manual and slow task. So there are lots of players in the industry who are collecting vast data sets of matched genotype and phenotype data, like Illumina, 23andMe, as well as state agencies like the 100,000 Genomes Project in the UK. But to this community, it should be evident that just collecting these vast data sets is not going to be successful in making accurate phenotype predictions, and that purely correlative models are not going to solve this problem. So the problem everybody is trying to solve is how to predict a high-order phenotype like a disease from a genotype. And while this community is very active of applying machine learning and deep learning technologies to this problem, it hasn't really taken a hold yet. And while every teenager is now using deep learning algorithms on their phone every day, we haven't been able to really get our work to be used in practice and benefit the lives of patients. So what's needed is that we build these what we call these deep biological architectures, because we cannot predict a disease directly from a genotype. So we need in between these models that predict molecular phenotypes, like protein binding, gene expression, alternative splicing, and so And what we believe is that it really needs a concerted and well-funded effort to take these models from the academic domain and really allow them to change medicine and affect the life of patients. So that is what the company Deep Genomics is about. We want to use machine learning to close the genotype-phenotype gap. And, we are located in Toronto. We've received $5 million in seed funding from some leading VC firms. The president and CEO is Brendan Frey. We're supported by very high-profile advisors from biology as well as machine learning, like Stephen Scherer, the director of Autism Speaks, Jan LeCun, the inventor of ConvNet and director of AI at Facebook, and Amshel Kondaji from Stanford. We're a team of 20 people now, growing to 30 by 2017. And we would be really thrilled if more people from this community would like to join us in this effort. And so please, if you are interested, then email us at hiring@deepgenomics.com or visit our website at www.deepgenomics.com/hiring. Thanks a lot.
Thank you very much for a good conclusion to this session.